Detection of cell cycle. Elabscience Cell Cycle Assay Kit is a kit that detects cell cycle by detecting DNA content. We have 3 kinds of dyes to meet your different experimental needs: sample preparation, culture cell according to the experimental design, collect the cultured cells. Centrifuge at 300 times gravity for 5 minutes. Discard the supernatant. Add 1 ml PBS to each tube. Resuspend the cells. Centrifuge at 300 times gravity for 5 minutes. Discard the supernatant. Add 0.3 ml PBS to each tube. Resuspend the cells. Staining. Store the absolute ethanol at -20 degrees centigrade overnight. Add 1.2 ml absolute ethanol from -20 degrees centigrade refrigerator. Mix fully. Store at -20 degrees centigrade for 1 hour or overnight. Centrifuge at 300 times gravity for 5 minutes. Discard the supernatant. Add 1 ml PBS to each tube. Resuspend the cells. Store at room temperature for 15 minutes. Centrifuge at 300 times gravity for 5 minutes. Discard the supernatant. Add 100 microliter Renars solution. Resuspend the cells. Incubate at 37 degrees centigrade water bath for 30 minutes. Add 400 microliter PI solution. Mix fully. Incubate at 2 to 8 degrees centigrade for 30 minutes in the dark. Detection.